Okay. Okay, to begin with, let me open the PyRx software. I open the PyRx software. Okay. Actually, the results will be saved somewhere else. I'll say where it is getting saved. But as of now, you put these files in your working directory. We call these folders as working directory. See, as wet lab scientists are saying as work lab, we will call our folders as working directories. So I have created a folder called docking. Okay. In the docking, kindly save the files, what I've shared with you. Done? Any doubt in it? Now, let me open the files and show it to you how it looks. So for this purpose, PyMOL will help us. PyMOL will help us to visualize the files. Even if you can't open in PyMOL, if you have Discovery Studio, you can open the same file and check over there. So we are trying to understand the receptor before docking, how the receptor looks. This is how the receptor will look. It's a beautiful transporter protein. Can you see the transporter protein here? Now what happens, our protein is going to navigate this path. Our ligand is going to navigate this tunnel. Let me check our ligand is entering here. Clear? So the step 1, file, load molecule, and load the receptor. Clear? So I have loaded the receptor. Can you see the receptor here? So can you see the XYZ direction below? Yes. The surface, what we have seen there. Can you see? Can you all see here? To make this visualization, it's very simple. You have to right click here, display and molecular surface. So people who can see this can give a thumbs up or a raise, hand raise. That is not in the manual, but for your understanding purpose, I have given this. Yes. Clear? Yeah. To make your visualization clear. Okay. Right. For some other purpose, we are going back to the display into lines itself. I am removing these balls and things. Why? Because I want to view the hydrogen. I am going to process it. So to view the hydrogen, I have just stopped it. Okay. Then right click. AutoDock. Make macromolecule. What I did? Right click. AutoDock. Make macromolecule. If you are feeling tough to follow me, I have shared the manual here. You can follow the manual too. Can you see here? Load molecule. Then what? You have to, the navigator button, you have to click make macromolecule and save in PDB-QT format. Save in PDB-QT format. Clear? Done? Have you seen some changes here? Previous structure and this structure, what changes you have observed? Can someone answer this? Out of the 24 participants here, yes, white lines, yes. What might be that white lines? Rather saying white, I can say some mild blue, cyan color, yes. Then none other than they are what? They are hydrogens. They are what? hydrogens. Clear? Yes. Yeah. Now once we confirm with our protein, see, I just want to use your mouse. Without mouse, you can't do bioinformatics work. So I'm just pulling in, pulling it out. Then now it's time for us to upload the ligand. To upload the ligand, you go to Open Babel. You have to click, see in controls, there is a button called Open Babel. Kindly click that. After entering to Open Babel, you just click over the, see there will be a small icon over there. Okay, that is called Insert New Item. Please click over there. Insert New Item. Once you have clicked over that, what you have to do is you have to open your ligand. Yes. Now ligand will be uploaded. Can you see the ligand over there? Everyone? As instructed in the manual, we have opened the ligand, correct? What's a follow-up step? We are going to minimize the ligand. What we are going to do? We are going to minimize the ligand. Clear? Yes, dear friends. Once you have achieved that, please listen here. Right click, minimize selected. Now it will ask for force field. Now it will ask for what kind of force field you are going to choose. How many of you are aware of force field concept? What is called force field? How many of you are aware of Newtonian mechanics? Do you know who is Newton first of all? Of course, everyone knows. If we know Apple, we know Newton, correct? What Newton have suggested us? What is Newton's second law? Am I asking very basics? Yes, F is equal to ma, correct? Right. So using Newton's second law, yes, exactly. Force field includes bonded and non-bonded parameters. Using Newton's second law, we are trying to solve many problems. Like we understood the entire atom as a single entity. So previously, scientists have had a confusion with Schrodinger equation. If you know Schrodinger, they tried to understand the motions of molecules. See, all the molecules are keep on vibrating. Okay, so bioinformatics is merely a thermodynamics application. Okay, we are trying to simulating the real-time situation. So in which what happens, we are defining a parameters. In what parameters the file should be there. Once we define the parameters, that is called force field. Okay, what kind of movement have to be there, what kind of the force should be there will be there. That will be mentioned in the force field. So let me have conjugate gradient force field itself. Okay, and I'm giving OK. Can you see there was a quick run over below? Below, can you see a quick run over? Now the energy have been minimized. As I said in the beginning of the class, minimized energy means maximum stability. Okay, now the molecule is minimized. Once I minimized, now I'm going to save that into a PDBQT format. So convert the selected into a PDBQT format. Can you see here? Since I have ran many molecules, it's all displaying all together. But in your case, if you are installing PyRx for the first time, there will be only one molecule. Clear? Yes. Now we will move to the next step. The next step is selecting one ligand and converting into auto PDBQT format that we have completed. We are setting the grid. We have to set what? The grid. How to set the grid? Yes, to set the grid, click over Vina wizard and select molecules. Clear? Vina wizard and select molecules. Where we have to select the ligand properly and the macromolecules properly. Clear? Then choose over the protein and the ligand. Once you have chosen both the protein and ligand, okay, yeah, then you have to click Run Vina button. After pressing Run Vina button, you have to press forward. That's it. Done? Any doubts in it? I'm ensuring that all the things are inside. All my surface are inside. I have to rotate in all directions. So for simplicity purpose, I'm removing it and later I can show into the display. And no need for extra space. Just cover in a proper space, proper surface. Okay. Once you are sure about the surface, please check, verify. Yeah, now it's fitted. Correct? Your voice is so feeble. Can you please be louder or keep your mic nearby your thing?
See, in the
Huh.
manual, we have given till using a open bubble, and you have been minimize selected and convert selected to AutoDock Ligand, right? PDBT
Correct,
file.
correct,
So
correct.
till that I did it, but after that I mean I'm not sure how to follow it.
Ah,
So
so okay.
it's there
Can you
I'm
share your
stuck.
screen? I'll help you.
One second, I just closed it actually. It's not coming, so I thought of doing fresh.
Okay.
An error has come. I just encountered some two or more
If
errors.
you want to see, actually, first 5 steps are so easy. If you want, you can do that first 5 steps and ask my help, and you can share your screen. We are ready to help.
Okay. No,
That's a real
you.
motto of this hands-on session. So
Yeah, yeah.
hands-on session means we can view via screen and we can correct your errors, troubleshoot your errors.
Okay. So once
If you
the for converting it into PDKPT file,
Hmm,
I got one ligand and one macromolecule, right?
correct, yeah,
And after that, I have to go to Vina Wizard and I have to
correct,
select the molecules.
exactly. Once you select the molecules, it may
And
have a
just
confusion.
selecting the molecule, how it has been done. That is by plus add ligand, add
No,
macromolecule.
no,
That is
no. You should not press. You should not add legends or anything. You just have to press over the above button. Can you see here?
yeah.
There are three buttons. Three things are there: navigators, controls, and view. Correct?
Um,
See, look at here.
yeah, yeah, yeah,
Look at
yeah,
my
yeah.
arrow.
Yes.
Navigator, view, and control. Correct?
Yeah, yeah. Mm.
So in navigator, you have to choose the ligand and a molecule. Both the things you have to choose from there, not by adding below. There is an option for add. Don't do that.
Okay. So I've
It's a bit
chosen
tricky.
one ligand and chosen the
Yeah.
macromolecule.
Yes.
After that, I have to.
That forward. There is a button called forward. If you choose properly, you will have a thing called forward. Correct?
Yeah.
If not, you won't get forward.
Okay.
It's same. It's we are doing exactly the AutoDock Vina using PyRx. That's it.
Okay. So
It's
once
sim
I clicked in forward, it's showing that ligand is accepting and it's
hmm.
asking for some maximize and reset. So once again, I have
Correct,
to.
correct. You have to adjust the grid like this. What we are doing now. You have to adjust the grid. Can you see the grid here?
Yes, sir.
Yeah, like this. You have to adjust the grids.
Mm.
If for your research article purpose, later you can note down this grid. Like what is the x-axis, what is y-axis, what is z-axis. In some of the articles, they will ask for these details, but not all the articles.
So once the grid has been adjusted, I have to do the forward again.
Yeah, exactly. No, you have to run Vina. Once everything's either run Vina or forward, both are same. So dear participants, we are running Vina. So I have 8 CPUs. So it's using all the potential of 8 CPUs and it's running.
Okay. It's running. Progress is running. Okay, I got it.
Thank you.
Thank you.
Yes, others, you can make use of this opportunity. If you have any doubts, please ask us. We are here to help you. We are here to help you. Can you all see this? So totally how many poses have came? So there were 9 poses. Okay, 0 to 8. In which pose is very good? The first one. So Dr. Anil have asked us a question in the beginning of the lecture. What is binding affinity and why it is important? So this binding affinity data will be acquired from here. Clear? So this binding affinity you can note down for your research purpose. Okay, once you're done with that, then we can go and view the files. To view the files, it will be stored in a separate place. That you have to achieve through edit, preferences. Listen carefully, everyone. Edit preferences and copy this workspace folder. So we are going to view the interactions in. Okay, we are going to view the interactions in PyMOL. So I am locating the folder. Actually, my folder is in desktop itself. So I can easily go through. Some of you might have in C drive some program files. So for them, the easy way to navigate is go to PyRx, click edit, preferences, and copy the path. Copy this path and put it over there. Clear? Put it where means put in the URL. Like click here and put it here. Enter. And enter into a folder called macromolecules. Any doubts, please ask us. And those who have achieved till this stage, like locating the folder, can raise your hands. Your folder should have the PDBQT format. There will be an out file. Okay, there will be a PDBQT file. So you should find that. Those who are done till this can raise your hands. Yes, great. What about others? We'll wait for your friends for 2 minutes. Please try to complete.
Hello, sir.
Yes, doctor.
I have a small doubt.
Yeah.
Hello.
Yeah, I'm hearing. Yes, please.
Sir, like yeah, during grid box setup, you have selected the whole protein, right, sir?
Yes, sir. I have selected the whole protein. Yes.
But, if we see in the PDV, like there is already a one inhibitor is there, no, sir, in this PDV.
No, sir. I have what the PDB I have provided is independent one. Should be independent one. 4ZY3A.
Okay, so what I'm asking is, instead of selecting the whole protein, is it possible to select only the part where that
Oh,
inhibitor is
okay.
bound?
No, have you selected from PDB database?
No, I have done whatever you suggested the same way.
Yeah,
But when
the
I
same way means already I have cleaned the structure. I can understand, sir. Like, the thing is, I already cleaned the structure. Okay.
Okay.
Those who are downloading from PDB have to remove the other ligands or other chains.
No, sir. What I'm asking is it possible to set up the box only in that region only where that inhibitor is bound?
Yeah. Definitely. We also can do site-specific docking. Yes, you are right. See, actually, we can do site-specific docking or ligand-specific docking. Can you see the structure here? See, initially we are worried where our ligand will bind. Can you see it exactly located in the gap?
Yes, sir.
But this is a blind docking. I have selected the entire molecule and the software have decided to be in the center. But you are aware, if you are aware of your ligands, what you can do is you can click over in the side where you can choose the ligands. For example, I have to click in this ligands, then I can choose those regions and I can set the grid box over there. By that way, I will do site-specific docking.
Okay, sir. Sir, which one is more valid?
But if you are aware, for example, in enzymes, if you are docking an enzyme and substrate, go for site-specific. If you are docking a receptor, better go for blind docking.
Okay, sir.
Because site-specific, no, people might be already reported a lot.
Okay, sir.
For blind docking, we can find for new targets. Like we can report to the reviewers like new targets have been found.
Okay, sir. Clear, sir.
Yeah, thank you, sir.